Okay, well, let's get started. It is unbelievably June already, right? And last time we had our exam, last week was sort of a weird week, wasn't it, for this course? We had a short week. All we really had was the exam. I only saw a few of you in lab. The rest of you are having lab 3 this week, so a little bit of an unusual week. This week we're back into the full swing of things with lab and lecture twice, and your exams went really, really well. I was very, very pleased with the exams. In my recollection, the highest average ever on exam number 1. And so the average was around an 83. The median was closer to an 85, which is really the highest I've ever seen it. So outstanding. Good work. I'm hoping that the new book is perhaps making a difference in this. So this is a new book. I think the book is more approachable. I think it's a more user-friendly book. And it'll be interesting to see if we can continue this high rate. I'll give you guys the credit and the book the second credit, okay? Because you guys are the ones working hard. But I was really, really pleased with that. Now, if you go ahead and look at your syllabus, let's take a look ahead. And I should say, too, your exams are available for review upstairs, Monday, Wednesday, and Friday afternoons, and Tuesday, Thursday mornings. Hillary's up there. She's got the key. She has your exam, and you can pick it up at that point. If you don't get in there this week, then next week I'll bring a part of it into class for you to pick up. But really all that you'll pick up is your vocabulary words and a page that says what your score was. But I do encourage that you go in and look. It's amazing what you'll learn about how you did during the exam. Even if you did well, go in and see how you can do better next time. And if you did lower than you expected, then it's always a very good thing to go in and discover what sorts of things you could do better for the next exam. Now the next exam will be what? Chapters 3, 4, and 5. Now, it's also going to include vocabulary slides, 21 through 40. And that exam is when? Next Wednesday. So I'm going to have to go a little bit faster through the vocab right now and give you about 6 slides per lecture between now and then, 6 or 7 slides. And you guys don't know this, but the chapter numbers are a little bit different in this new book than what they were in the old book. And so as you're looking at your PowerPoint notes, I'll make a couple of corrections for you. But chapter 3 is all about cells and what we'll be discussing today. On Wednesday, we'll get over to talking about some histology, which is what we did in lab this week and last week. And then next Monday, we'll be discussing skin. And those 3 topics will be your content for exam number 2. So let's get started with that. And again, we need to get through 40. So I need to go through 20 slides. I'll go through I'll go through 6 or 7 today, 6 or 7 on Wednesday, and then again next Monday. Definitely make your note cards up for vocab. Don't wait for me to go through this. You can go ahead and make up those cards. Going through 40 means that we're going through what term? Through slide 40 means that you can go ahead and make your vocab cards up through hystero, uterus or womb. Okay, so go ahead and make those cards up, then you'll be just ahead of the game. So let's get started with this. The vocab went really quite well. I only have a recollection of 2 or 3 students for whom the vocab seemed to be a foreign language. So that's really good. So if you did not do well in the vocab, please, now you know what to expect. It is going to be a fill-in-the-blank kind of thing, and you just have to know the terms. And if you'll work ahead on those, I don't think you'll be stressing about it. If you wait till the night before the exam, it's usually not a good outcome. So don't let that pile up too much. Go ahead and get the vocab going now. Okay, so vocab, 21, cutie, cutaneous, cuticle. Right, the skin that's on your nail. So cutie means skin. Cyan, one of the color cartridges in your color printer. Cyan is the bluish color ink. Cysto, sac or bladder. A cyst, right? A cyst would be a sac or a bladder or a fluid-filled container. Cyte and cyto, both referring to the cell. Dactyly. Fingers, digits, or toes. So polydactyly would be a person who has 6 or more toes or 6 or more fingers. Maybe you have a cat that has polydactyly. Syndactyly would be where two fingers are joined together. Syn, where they're brought together, so that'd be a webbed finger. D, down from or to undo. So to defibrillate, if someone needs to go under defibrillation, fibrillation is not a good thing. Fibrillation is an uneven heartbeat. So to defib is to stop the irregularity and bring it back to a normal rhythm. Dend, tree. When we get to the neurons in the nervous system, we'll talk about dendrites. And dendrites are these little tree-like branches off from the neuron. Demi, like semi, half. Okay? So demi or semi, both meaning half. Derm, skin. Derm, go to the dermatologist. You'll hear a lot of terms, hypoderm, epidermis, dermis. Detruse is to force away. The muscle in the bladder. One of the muscles in the bladder is called the detrusor muscle. And as you void, as you urinate, you're pushing away the urine with the help of this muscle. Dextro is to the right of. Dextrose, sugar, common sugar, is a, quote, right-handed sugar. Anyone know what left is, the opposite of dextro? Yeah, we'll see L in the chemistry, but it also is S for sinister. Sinister is left-handed. So if you're left-handed, maybe you're sinister. So we'll see that term later on. Di is two. Okay, that was not so difficult. And dia, across or through. Diameter, right? The distance across. Dialysis, when you are moving something across a membrane or through a membrane, your kidneys are filtering, and we can do dialysis to make up for what your kidneys cannot do. Diastole, the time when your heart is not beating, the time of relaxation, the time standing apart between the contractions is the diastolic pressure. So diastole, the relaxation, or the time in between, the stand-apart time. Dips, thirst. If you have polydipsia, you are incredibly thirsty. Polydipsia. Dis has a number of different meanings, and I think something they'll speak to us here. We've got apart, or opposite, or absent. So to dissect, to pull apart. To disinfect, the absence of infectious agents. Disarticulated skeleton. Next week, actually this week, Wednesday, we'll be playing with bones. And some of the bone sets you'll be playing with are in a box. All those bones are disarticulated. They're not touching each other. So articulation means to come together. Disarticulated skeletons, ones which have been pulled apart, if you will, and or taken apart, right? So apart, opposite, or absence. And then dipple, double. A diploid cell. Most of the cells in your body are considered diploid. That is, they have a double set of chromosomes, or they have two sets of chromosomes. Diuret, to pass urine. Diuretics, medications one takes to encourage urination. Dorsal, think dorsal fin, toward the back, we know that one. Duct, to lead or to draw. So think ovarian duct, and what's going down the ovarian duct, which is also called the fallopian tube, is the egg. So the egg is being led down the tube, if you will. Also, though, if you are to do adduction, adduction or abduction is to move or to draw or lead your body in one particular direction. Dur, hard. When we get to the spinal cord, we'll see the dura mater. It's the outer hard layer around the spinal cord. Dynia, pain. Dynia, pain. Dys, also pain. So dysuria would be painful or bad or difficult urination. So the dys, bad, difficult, uria, urination. E, out from. So think exit, right? EX or E, out from. And eel, pertaining to, we've already had ac, pertaining to, and now we have eel, ac and eel. And I'll go through this last one for today. EF, away from. So E or EF, away from. Efferent, we'll see that word a lot in about a week or so. It means to draw something away from. Ecto is the outer layer or the outside. ectomy to cut out, so appendectomy to cut out the appendix. And finally, if you have swelling of some sort, you have edema. So edeme, referring to swelling. Again, we'll go all the way through 40. So go ahead and get those note cards going. You know what to expect now. It is non-cumulative, so I will not pull terms from the first 20. Okay, so it's just going to be the subset from 21 through 40 on the vocab. Again, going through to histero. Now on Blackboard, I have renumbered this PowerPoint, and I'm calling it 3B. So please, you can do the same thing. This does come from my previous textbook from Chapter 4. But for the new textbook, and as I get this turned over, this is like the second half of Chapter 3, okay? So change this to Chapter 3B, or the second half of Chapter 3. In the last discussion we had, and I know it's been a while, it's been a whole week before the exam, or actually it's been even more than that, right? Quite a ways back. And we discussed the cell, and we looked at the different organelles, and discussed the Golgi and the lysosome and the peroxisome, and also spent quite a bit of time looking at the plasma membrane, talking about how some molecules can get across the membrane, others can't. Is there anything from that conversation that needs to be clarified? You will find mastering homework assignments available for you on chapters 3, 4, and 5. Apparently, it's not as easy as I thought for you to be able to go back and do the homework assignments multiple times. So I will do what I did last time, and that is I will repost them. And it's kind of awkward, I know. But you can do them the first time and the only time if that's what you choose. But if you want to do it a whole second time, I'll simply post it a second time. If you make a higher score the second time, that's the score that I will capture. Okay. So I apologize. There's a little bug in there that I haven't quite figured out why you aren't able to interact with those multiple times. Even though I've told it that you can, it's not allowing you to. So anything on organelles? Does that conversation make sense? Know the organelles, recognize them, label the parts of the cell. You can definitely expect a PowerPoint image on exam number 2 where you'll have to identify and know the function of different organelles within the cell. Continuing on with chapter 3, I should say, is more about what the cell does, some of the special processes that the cell's doing as a part of its cell cycle and as a part of its well-being. And we'll pick this right back up with this slide. You saw this at the end of the last presentation, and this is a chromosome. Okay, on the right-hand side, we have a chromosome. We see that coiled DNA, right, all this. And what we see under the microscope is this letter X-looking structure. And when you're looking at cells undergoing mitosis, it almost looked like cross-stitch, little X's. And we are reminded that this chromosome is actually DNA, but not just DNA, but DNA that is wrapped around proteins. So collectively, chromosomes are not just DNA, although that's the first thing that comes to our mind, that it's this really, really long molecule of DNA, but this DNA is wrapped tightly around proteins. So collectively, we call this chromatin. Okay, chromatin is the combination of DNA wrapped with the proteins, and that word is kind of hidden over here, formatting issues. But you'll see the word chromatin in there. And this is highly condensed. That is, it's visible, right? When we say that the chromosomes become visible, another way of saying that is that the chromosomes condense. They become so dense that we can see them under the light microscope. So what is a chromosome then? Again, it's the most organized level of your genetic material. And as humans, how many chromosomes should we have? How many chromosomes total should we have in our cells? Each cell should have, am I hearing 46? Okay, so each cell should have 46. 23 that came from mom, 23 that came from dad. Now, when those chromosomes came from mom and dad, they were coming from their egg and sperm, and there's another process that occurred in making that DNA, and there was a shuffling, a rearranging of the genetic material, so that it's a random act, right, with the genes that come in from the sperm, those which come in from the egg, and here we are as a result of that DNA. Again, each chromosome then, what you have in each one of your cells are 46 really, really long molecules of DNA that are wrapped around proteins. And that wrapping around keeps them shorter and helps them to stay organized. We only can see the chromosomes, again, during mitosis, that is when the cell is actually dividing. Now, the cell cycle was described a little bit in lab. You know that every cell is somewhere in its own cell cycle. And we have trillions of cells, million cells, right, in our body. And those cells, each of them is somewhere in its own cell cycle. We have to be continuously making new cells within our body. We get that. We just don't think about it, I don't. We know that we get bigger. We know that we're once young and now we're adult size, and we know that with time, likely we'll lose some muscle mass and we'll lose some of our height. So there's this constant change of cells. We're also making blood cells all the time. We're renewing those blood cells. We're losing skin cells to the vacuum cleaner constantly, and we're replacing those cells. So there's a constant replacement of cells, not only as a normal process, but to replace damaged cells. So not only do we have apoptosis, remember that term? Apoptosis is what? I mentioned it in passing. Yeah, programmed cell death that's supposed to happen. And so that's definitely going on. But then there's also the chance that you cut yourself. And as you cut yourself, that wasn't necessarily planned, but you cut yourself, and now the body has the ability of replacing or replenishing some of those cells. Some cells can be replaced. Others cannot be, as we'll discuss more. Some cells, like your heart muscle cells or many of your brain cells, once they are formed, there's no mechanism for them to replace themselves. So that's why stroke and heart attack are so devastating, is because those tissues do not regenerate themselves once they're damaged. But you cut yourself, burn your tongue, and you know that, hey, within a couple of days, everything will be replaced and life is going to be good. So again, that term apoptosis is programmed cell death, part of what's supposed to happen for your overall body's metabolism. Now, as we go into this conversation about the cell cycle, I know that it seems rather academic. It's like, do we really care about these cells and what's going on with them? But I'm hoping that by the time I finish this little slide, that you'll be at least a little bit more intrigued by this. So we'll start off here with the reproductive system. We'll come back to the reproductive system in the very end of the book. But what we have here is the uterus, and the uterus, here's the vagina coming up to the cervix, the narrow opening into the uterus. And going off both sides are fallopian tubes or oviducts, and we see one of them here. At the distal end, at the far end of the fallopian tube, here's the ovary. And the ovary is going to, typically on every other month kind of cycle, release an egg, and that egg will be drawn up into the fallopian tube, up here. Now, there's a possibility that sperm are there, and the sperm are viable for about 3 days. And so sperm could be there already or could arrive right after the egg is released. The egg is only viable for about 24 hours. So the egg has to be fertilized within 24 hours for normal fertilization to occur. So normally the egg and sperm are going to meet each other up here toward the very top of the fallopian tube. And then as the egg and sperm come together, and that's what we see at the very top picture, egg and sperm coming together, and we form the zygote. And the zygote is the coming together of the egg and sperm. That's fertilized egg. And then that cell will undergo cleavage. It will divide. And we'll have 2, then 4, then 8, and on it goes. And we'll reach a solid ball of cells called a morula. You'll see that term again. Don't worry about it right now. we'll come to a solid ball of cells, and then eventually we'll come to a hollow ball of cells. Well, that's called a blastula. Well, that's the same state we were looking at with the fish last week. When we looked at fish blastula, it was a fish blastocyst or a fish blastula. It's a very young embryo, and we looked at cells in that ball of cells undergoing mitosis, right, as an example of seeing some nice ideas of mitosis. It takes about 5 or 6 days for this journey to go from the fallopian tube down to the uterus. 5 to 6 days. And during that time, this cell is dividing very, very rapidly. Right? So by the time the embryo reaches here into the uterus, it has reached a size and a state of development, whereas it can continue. It can embed into the uterus wall and maintain shop for the next 9 months. If this embryo has not reached a sufficient developmental stage, right, within those 5 or 6 days, it won't be able to adhere and hang on to the uterus. And so that embryo would be lost, right? It would never adhere to the wall and would simply be passed. And the woman, certainly fertilization occurred, but pregnancy did not occur, right? So we know that fertilization happens far more often than pregnancy. Pregnancy is the implantation of the embryo, whereas fertilization is simply the coming together of egg and sperm. Okay. So the cell is in rapid division, isn't it? It has to do this rather quickly, because if it doesn't get to a sufficient state of development, it would never be able to establish pregnancy. Now, sometimes the fallopian tube can be scarred, or for whatever reason, the embryo gets stuck in the fallopian tube, and it can't get beyond a narrow area, and this is called an ectopic pregnancy. Now, ectopic pregnancy tells us that the embryo is in the wrong place. There's no way that this embryo can survive there. It can survive for a couple of days, but it's eventually going to start causing pain as it enlarges, and it will never be able to stay there through its development. So that can be a very serious situation where the fallopian tube can actually get ruptured and cause even more damage and more scar tissue in the future. So those have to be surgically removed, those ectopic pregnancies. So again, I hope you appreciate that. Oops, we have a rapidly growing ball of cells here called an embryo that has to get to a certain point of development in order for fertilization to occur. So the cell cycle is important to early developmental biologists trying to understand this cycle. But where else do we see rapidly growing cells? In a not so good situation. Cancer, right? So we'll tie this in with cancer here in a moment. Now, these cells are rapidly growing. They're undergoing rapid division, and we know that that term is mitosis. Now, mitosis is just a part of the cell cycle. Remember in lab we mentioned the whole cell cycle, and I'll show you a picture again in a second. But there was the G1, the S, and the G2, right? And that was the rest of the time that was collectively called interphase. Interphase. So inter meaning between. Inter means between. So interphase is the phase in between all of the mitoses, all the cell division periods. And we also saw that mitosis is a highly visual stage, that you can see cells undergoing their little division dance, if you will. And so mitosis has been divided up into other stages or into subdivisions. So we have prophase, metaphase, anaphase, and telophase. And we went over that in lab, and I think we've got this pretty well down. Now, the purpose of mitosis is for that one cell to make two identical cells like itself. When mitosis is over, when the cell actually completes the formation of two cells, that moment is referred to as cytokinesis, and that is the making of the two daughter cells. One way to remember this is simply, I passed my anatomy test, right? So interphase, not as a stage of mitosis, but as the stage beforehand, and then prophase, metaphase, anaphase, telophase. So here's a reminder of that cell cycle. I've got 3 or 4 different pictures of it here. Here, it looks as if mitosis is about 50% of the cell cycle. That is not an accurate representation of the time. Okay? Cells can be in interphase for months and months and months, and then divide very, very quickly. So time-wise, right, mitosis is not 50% of the overall cell cycle. But because we know more about mitosis, and because we want to divide this up into its individual spaces or its phases, you'll see it stretched out here in this.
Can I talk to you for a second on the whole?
Yep.
Just real quick.
Okay, so what we see here is that the cell cycle is this cycle, but time-wise, right, this is not a good, accurate description of time. So we do want to go back and briefly look at prophase, metaphase, anaphase, and telophase. I think you've got that. We've talked about it. You've been quizzed on it. I think this is coming along pretty well. And then we see that the rest of interphase, which is shown here in blue, is the G1, which some books call growth, some books call gap. Either way, gap or growth phase. Then we get into S phase. Now, I'll come back to S phase in a few minutes. S phase is a specific time, S standing for synthesis. And it's during the synthesis phase that the cell is undergoing replication. And replication is a copying of the DNA, making a replica of the organelles and a replica of the DNA. And then we get into a second growth phase. Again, all of that is interphase. Now, what can we see in the cell during interphase? If I take a look at these cartoons, what do these cells look like? I see a nucleus, right? I can see a nucleus, but I can't see individual chromosomes. So I see I just see a nucleus, and I also see, what are these guys? These 2 green guys. Centrioles. Each one is a centriole, and notice that they're kind of clustered together. And during mitosis, what's going to happen? They're going to migrate to the north and south poles of the cell. So during interphase, we see a full nucleus. We cannot see the chromosomes, and we notice that the centrioles are sitting together in an area of the cell. So quickly, on the left-hand side, again, I see a nucleus. I can't see individual chromosomes, and I see the centrioles together. So this is just good old interphase. I'm not going to ask you to recognize interphase. Okay. I do want you to know the other phases, though, of mitosis. So starting here, the nucleus is gone, isn't it? The nucleus is no longer a nucleus here, so the nucleus is gone. The centrioles have moved to their respective poles, left and right side. The chromosomes are now visible to me, now visible to you under the light microscope. Now remember that these chromosomes have already duplicated, haven't they? They did that before they went into mitosis. They already copied themselves. So when we go into mitosis, we already have 2 of everything. So that when we divide here in a few minutes, we get 1 copy of each molecule to its respective cell. Then go ahead.
How would you recognize those on a slide view 1, 2?
The centrioles?
Yeah, like I see the nucleus, I'm assuming is the very dark shaded area here.
The whole center is a nucleus. Now the dark center is a nucleolus.
I mean the bottom one for the actual slide where you're showing.
Mm-hmm.
Can you see the centrioles in the actual?
No, no. The cartoons are much clearer, okay, when it comes to these organelles. So no, you cannot really make those out. I agree. Now, yeah, no, you cannot see those clearly at all in the bottom picture.
I just, the thing that kind of looked like little spiders on each side, I just wondered if,
In here?
there and then over here.
Yeah, don't try to read too much into that,
No.
okay? Now, the next three I think we've got down pretty well. So if you see the chromosomes in a nice straight line, you're thinking what? Metaphase. Now, what has happened? Also, in addition to lining up, from the centrioles, what has happened? Mitotic spindles have moved out, and they're going to grab onto each of the chromosome pairs. And then during anaphase, when the chromosomes are being pulled apart, right, they're retracting. And then eventually, we have the cleavage going on. And you can even see in this cartoon that the nucleus is reforming. In the actual micrograph, you can see the cleavage. You can see the complete separation of the chromosomes. Okay? So if you just know the stages of mitosis, and then recall that I told you what's going on in prophase. So let's go back and make sure. What are the three things that's happening here in prophase? Yeah, the spindles or the mitotic spindles are what? Deforming or elongating, or visible, however you want to think about it. But they are being formed. Number two. What's going on with the nucleus? The nucleus is going to break down, dissolve, disintegrate, right? So we're going to lose the nucleus, disappear. And then number three, what else is going on? The chromosomes are becoming visible, and the word I'll use for that is that they condense. Right? So we know that the DNA is wrapping around those proteins and becomes so densely packed that we can actually see it. Now, mitosis is not the only way that cells are dividing. It is the most common way in your body. Mito, literally from the Greek, means threads. And so the scientists didn't know what was going on, but when they first were able to see cell division, they saw threads. Well, we know that those threads now, like that cross-stitch pattern, those are the chromosomes. So it's during mitosis that we can, quote, see the threads, right, these chromosomes. But there's also meiosis, and you hear meiosis as well. So meiosis is another type of cell division. And in this case, you're in the process of making egg or sperm. So meiosis is only used in creation of the sex cells. What's the name for those sex cells? Collectively, the gametes, right? Egg and sperm collectively are the gametes. And the gametes to be made depend upon meiosis. Another way of saying it is that mitosis, the regular type of cell division, is for the making of all of your somatic cells. Now, soma is a term we'll see over and over, but soma means body. So your somatic cells are all the cells of your body, your typical body. I mean, every other cell in your body is a somatic cell. Now, again, I know that this may sound really, really academic. Well, who cares about meiosis? Well, meiosis is, again, the important process in making egg and sperm. It starts off, though, with a regular old cell. So meiosis begins with a diploid cell. We know from our vocab, a diploid cell is a cell that has two of all of the chromosomes. And in the final process, or the final product of meiosis, is to create a haploid daughter cell. And haploid means a single set of chromosomes. So one set of chromosomes. At this point, we have egg or sperm. And there's that word gametes. Okay, gametes, sex cells, or sperm or egg. Now, meiosis is critical in gametogenesis. Gametogenesis. Any idea what that word refers to? Yeah, genesis in the beginning, the formation of gametes. Right? So the process of making sperm or the process of making eggs, collectively, those are the processes called gametogenesis. More specifically, gametogenesis we can break down into oogenesis, right, the making of egg, or spermatogenesis, the making of sperm. Okay, so oogenesis or spermatogenesis. So why do we care? Well, maybe you know a person who looks a lot like this young lady. All right, this is a child with Down syndrome, and Down syndrome is the direct result, 95% of the time, is a direct result of something going wrong in meiosis. So what's the other name for Down syndrome? Who knows the other name for this? Yeah, trisomy 21. So in this case, these individuals have not 2 chromosomes of every chromosome, but they have a third, number 21. It just happens to be that it's a rather small chromosome. And what happened is that during egg or sperm production, there was a nondisjunction. Nondisjunction is a fancy way of saying that when the chromosomes were pulled apart in anaphase, they didn't separate properly. And one of the daughter cells got both copies of that chromosome, and the other cell got none. So looking over here on the bottom, this particular non-disjunction event happened. You see the chromosome being pulled apart here? These two traveled together. And so the cell, one of the gametes, ends up with an extra 21st chromosome. If that was the egg or that was the sperm that was used to create the zygote, this young person, then that individual would have trisomy 21 or Down syndrome. Over here, this egg or sperm is lacking one of the chromosomes. It doesn't have a chromosome 21. If that egg or sperm was used, then what would happen, do you think? Any guess? It's non-viable. So as humans, as mammals, we have very little wiggle room when it comes to extra or missing genetic material. If we're missing a chromosome, it's not going to be life. Now, there's one exception to that, and that is some women are lacking a second X chromosome, but that's a special case. But typically, if you are missing a chromosome, you're not with us. Okay. And just a little bit of extra genetic material, like this situation where there's an extra chromosome 21, leads to a whole series of issues that we call Down syndrome, described by a guy, right, Down, who described patients who all had the same characteristics, and now we know what's causing this. So they have heart issues, they have mental retardation to some degree, they have a unique look to them, right? There's a very unique facies, if you will, for kids and individuals with Down syndrome. It's not specific to any race. There are people with Down syndrome all over the planet, and they all sort of have the same look to them. Wonderful, wonderful folks. But you may know someone. So that's just a problem with Down syndrome. I tried to tell you last time, too, why do we care about lysosomes, or why do we care about peroxisomes? Well, when those organelles are deficient or defective, there are diseases that are associated with those as well.
That's in both male and female too. Like I guess I always thought that Downs was just solely the egg, but it can be.
It can be the male. Now, it is typically, it is more often than not the female. And that'll make more sense later on, but it can happen in the male. Absolutely can. The difference is women are born with all the eggs they'll ever have. And so the eggs are 20, 30, 40, 50 years old, and it turns out that the aging of the eggs can certainly contribute more to this non-disjunction event. But it can certainly happen in males. Absolutely. Now, are there other trisomy examples? There are. Trisomy 21 is the most common. Most of the other trisomies are fatal. So the extra chromosome is not consistent with life. There are a couple of other ones, trisomy 15, trisomy 18, and in those situations, the individual is far more troubled. So life is much more difficult. As far as all the trisomies, 21 is the most common and the one that is most tolerable, if you will, for life. So these folks survive quite well. Let's talk about apoptosis as well. So what I've got here is a human embryo. We're going from week 5 to week 10, okay, of embryologic development. So here's 5, going over to week 10. And what I want to show you is at week 5, the hands are described as being paddles. The fingers are not individually formed. And then as you look across, by the time we get to week 10, you got perfect little toes, right, formed on this embryo. Little hands, little fingers. Now what happened during this period is apoptosis. So the hand was a paddle, and certain cells knew to die to separate those structures into fingers. So the bones were formed separately, the skin was separated, and that's just a really cool example of apoptosis. Now, if for some reason apoptosis does not occur properly, and the fingers are maintaining their wedged appearance, then that person would have what?
Syndactyly.
I mentioned dactyly being fingers or toes. Syndactyly, as the example was, when they're webbed together. So a person would have webbed fingers or webbed toes if apoptosis did not occur properly. So it's interesting. Cells know when to die. Cells know when to divide, right? Cells know when to create more of themselves. If right now, it's kind of an odd thing to think about, but if every cell in my right arm decided to undergo cell division right now, then my arm would be twice as big, or maybe if it was twice as long, I could just be scratching my toes right now without bending over. So, but it's cool, right? Our cells know when to divide. They know when not to divide. There's a cell cycle that is well controlled. We are largely symmetric individuals. Our right and our left legs and arms are pretty much the same size. Every cell in your bones knew to grow to a certain shape. And every bone in your body has a certain shape because it has a certain function. Your femur, right, the big bone in your leg, is much, much larger than the little bones in your ear, the little ossicles, or much different than the other bones. So, I mean, every bone has its own shape. It knows, right, those are cells. They're living. So these cells know when to divide, when not to divide. So now let's describe when the cell cycle goes out of whack, right? So we're all living miracles, if you will, in that the cell cycle is behaving. But there are examples that we know about where cells no longer exhibit normal control over the cell cycle. And a tumor is a good example of that, okay, a cancer. And so basically, a tumor is when cells are growing out of control, when they're growing beyond their normal cell cycle. So we disrupt the balance, and cells are multiplying faster than they die. And we get a solid tumor, right, or a solid mass. And this can be referred to as a tumor or a neoplasm, new growth, neoplasm. And I won't spend a lot of time on this, but just enough to, again, appreciate why do we care about the cell cycle? Well, it's cancer when it's wrong. So we study the cell cycle to try to beat cancer. Because if we can control the cell cycle, then we can slow down the growth of abnormal tumors. So we'll start off with just a very basic idea of benign versus metastatic. So a benign tumor, we know what does Benny mean? Good, right? So if you're going to get a tumor diagnosis, you want it to be a benign. That's a good diagnosis usually. So a benign neoplasm or benign tumor is one that typically grows slow, or at least it is confined to a space. It does not spread. Okay, so a benign tumor is slower growing, or at least it is contained within a connective tissue capsule. The cells here are said to be dedifferentiated. Now let me describe that to you. Think about going back. I'm going to go back about 5, 6 slides, maybe more than that. Going back to the uterus, to make this point.
Ask a quick question.
Yes?
Can a tumor be a result of trauma?
Can the tumor be a result of trauma? No. All cancer is genetic. It didn't used to have that definition, but we now know that every cancer is the result of a genetic change. And that'll make more sense as we finish up this conversation. But no, tumors, cancer will always be a genetic change in the cell, not due to some trauma. So here's what I'm trying to say as far as differentiated. When you look at the zygote has the potential to become every single cell in your body, right? It's going to differentiate, and as it becomes 2, 4, 8, 16 cells, at some point, some of those cells will know to become muscle. Some of them will know to become kidney, liver, brain, stomach, you name it. And we say that the cells are becoming different from each other. They are undergoing differentiation. What we see in a tumor is that the cells have become de-differentiated. They basically have reverted back to a more adolescent or childhood form. Now, cells grow faster, right? Undifferentiated cells grow more quickly. That embryo, this blastula, is growing very, very fast. These cells are undifferentiated. They don't yet have their identity made. Every cell in this morula, every cell in this 8-stage set, I could take one of those cells and create another whole individual, which is exactly what identical twins are, right? At some point early, that embryo splits. And it's not like we get half a person. We get two now totally identical individuals. And so those cells have not done any differentiation at this point. Okay? Just want to get that idea of differentiation down. So what I'm telling you is that in a tumor, the cells are de-differentiated. That is, they have become less specialized. And they get their own blood supply. It's a ball of cells, right? It's a cell. We don't think about it, but cancer cells need a blood supply too. They need an oxygen supply. And many of the tricks that we do medically to kill cancer is to stop it from dividing, stop it from getting a blood supply, stop it from getting nutrients. But cancer cells have their own way of increasing their own blood supply. Now again, typically these cells or these tumors are not fatal unless they're pushing up against a vital organ or they can't be surgically removed. So a benign tumor in the brain, while it may not be spreading, if it enlarges enough and it cannot be surgically removed, it could become fatal if it's pushing against important nerves or blood vessels. Now I want to compare benign with malignant. Now malignant tumors are not what you want to hear about, and here's the difference. They are unencapsulated. Basically, they don't stay put. They don't stay within a little connective tissue capsule. And these are also de-differentiated, so they're a more simplistic, AKA faster growing version. They too increase their vascular supply. They need their own blood supply. They need their own route of oxygen. They grow very rapidly, but the real kicker here is that they undergo metastasis, or they metastasize. And that's a way of saying that they move or they spread to places they weren't normally, where they didn't start, I should say. So a tumor that metastasizes sort of breaks off. It's not being maintained in a capsule, and it spreads through the blood or through the lymphatic fluids to a distant, faraway place and sets up another tumor. And we know that some cells, some certain cancers, tend to spread to others. So some individuals will have a lung tumor, and we know that lung tumors oftentimes will metastasize to the bone or to the brain. And other types of cancers will, unfortunately, metastasize, sometimes in predictable ways, to other organs.
That's what I was just gonna ask you, is that why they say, where did it
Absolutely. But you had to have had lung cancer first, okay? So you may have a lung cancer that was a lung cancer, then metastasized to the bone. And when they do the biopsy on the bone, they're able to recognize that, yes, the cancer's in the bone, but it originated as a lung cancer. So then they can say, okay, it definitely originated there. The lung was the primary place, the bone was the secondary metastasis.
What's the difference in, I mean, how can they differentiate between?
Well, even though every one of your cells, right, every one of your cells, your lung, liver, brain, kidney cells, all have the same DNA in them, your body also tells, I mean, you have kidney cells and brain cells and stomach cells that all have certain jobs. How does your body know if it's a liver cell or a kidney cell? Well, there are certain genes turned on, certain genes turned off in that cell. And so the kidney cell has a particular set of jobs to do, and a liver cell has a different set of jobs to do. And those differences, right, are genetically, we can trace those genetic differences and determine is the cell originally a liver cell, a brain cell, or a liver cell. Yeah. It's really, really interesting stuff, unless it's your family member who's dealing with this cancer. Now, cancer, again, is a loss of control of the cell cycle. The cells are growing out of control. What color is cancer? In your mind, what color is cancer? Black. But on a biopsy, cancer is beautiful, new, glistening cells. Right? I mean, cancer are fast-growing cells. They're not old and ugly and black like we want to imagine them to be, right? But cancer cells are new, fresh, young, fast-growing cells. But we still have this image that they're black and ugly, right? Because they're damaging us. They're not a good thing. Okay, but if you were to see a tumor, you'd be impressed by how young and glistening and fresh it looks, if you will. So one of the unfortunate things are, how do these cells spread? Well, they lose what's called contact inhibition. If I take some normal cells out of from your skin, and I put them into a petri dish in the lab, those cells will grow. And they'll grow usually until the plate is filled. And when the cells begin to touch each other, or when they reach the edge of the petri dish, they stop. They know that when they touch each other, that's the limit of their growth. Cancer cells forgot the rules. So cancer cells, when they reach the edge, start to climb on top of each other. Right? They lose their contact inhibition, and they begin to make a jumbled ball. So again, it's another genetic defect in these cells. So they are growing when they shouldn't, when they start to overgrow each other, and they don't stop that division like a normal cell would do. They also, again, are de-differentiated. They're less specialized. They're growing more quickly. These cells release chemicals which encourage angiogenesis. Again, these cells need their own blood supply, so they're going to encourage their own blood vessels to be formed. Angiogenesis, we should be able to break that one down. Again, genesis in the beginning, the making of, and we know angio means again, they metastasize. That is, they leave, they jump off, and they travel to places they would normally never go, and they become more invasive. They have a tendency of squeezing into places and traveling where most cells would not go. And that is this term metastasis, right? The cell or the cancer has metastasized or is a metastatic cancer because it is spreading to places it would not normally go. Questions on cancer or the cell cycle? Do you care a little bit more now? I mean, I know this cell cycle thing seems so dry. Who cares? But this is what cancer biologists are studying, right? They're trying to figure out how to stop the madness, stop the out-of-control growth, and what can we do to interrupt the cell cycle? One and then two.
Is there anything in, like, as far as the organelles that is causing cancer? Like, there were parts of the organelles that encouraged splitting and...
Some organelles are definitely involved in cell cycle. So, for example, if a cell did not have centrioles, it couldn't divide. Okay, so is there one cell that I've said can't divide? I may have said it in passing. Maybe I haven't said it. Neurons, cells of your nervous system, don't have any centrioles, therefore cannot divide because the centrioles are what spit out the mitotic spindles to pull the chromosomes apart. So if you do not hear about cancers of neurons, because that kind of cell cannot divide. Okay, you get a brain, you get damage to your brain, and the neurons cannot regenerate. That's why brain damage is so devastating. But you also can't have a cancer of a neuron, because those cells do not have the equipment to divide. But ultimately, all cancer is genetic. There's a problem at the genetic level, at the DNA level, that's caused it to go out of control. So some organelles could contribute to it, but it would never be the cause. Question?
I'm glad you did bring it up because it is very interesting. When the physician wanted to give my 11-year-old daughter an HPV shot, and she said the way that they determine if they have HPV or if they have cervical cancer is the growth rate.
Absolutely.
And I never did understand. I'm like, growth rate of what? But now it's all making sense that you said that.
Right.
I was shocked they wanted to give it to her at 11.
Yeah. 11.
Yeah.
Those are general guidelines given for all population, but yeah, it's shocking to us as parents, but yeah, that's when they were targeting the kids. And they stopped targeting after 26. So 11 to 26, I think those are the guidelines now for HPV. So what we want to, as low as 9, yeah, but we want to make sure that everyone is immunized before any sexual contact. And we assume that by 26, everyone has had sexual contact. And so basically the guidelines are 9 or 11 up to about 26. Just statistically, numbers are established. Now your individual case may differ from that, but that's why those numbers are there. Okay, so that's cancer and a little cell cycle, a little mitosis. Yes?
So what is it that these cancer drugs that are out there are doing? Are they attacking the...
All sorts of things. So you've got some drugs that are blocking angiogenesis, the blocking the making of new blood vessels. You've got some medications that are interrupting the microtubules, which are the mitotic spindles. You've got some molecules that are, again, interrupting most of its cell cycle or blocking the advantage of the cell to move or migrate. Yeah.
Unfortunately, that does that to normal cells.
Well, that's just it, right? So cancer cells are just like the rest of our cells. They've got a problem, but when we give chemotherapy to a patient, we're not only trying to, we're trying to target the cancer cell because it's fast growing, but we also have to recognize that every other cell in the body is also being negatively affected. So you have all the symptoms and the problems of side effects of chemotherapy. And we'll talk more about that in a few minutes. Now, on exam 1, there will be a small written portion. And this is that written portion now. So I'm giving it to you plenty of time in advance for you to explore this. And these pages are not legit, so don't worry about the page numbers. I'll give you some new page numbers. But, and this is going to be on exam 2, not quiz 1. So again, I got some updates to do. But this will be on exam 2. And here's what you'll be asked to do. You'll be asked to write 3 sentences. And those sentences must include the following terms. So here you have 11 terms. You must use 10 of the 11 in sentences that make sense. If you turn to the back of any chapter, any textbook, you'll see some summary statements. And those summary statements are usually just densely packed. Right? All those major vocabulary terms of the chapter have been put into just a few key sentences, or maybe a couple paragraphs. And that's basically what you're creating here is a summary statement. Now the words here, some of these words you already know. For example, I've already said nucleus, you know a little bit about DNA, you know a little bit about the cytoplasm, and you've heard about the rough ER, ribosomes, and proteins. In passing, you've heard about those things. And you could create a sentence right now, perhaps that used ribosomes, proteins, and ER, rough ER. Can you imagine a sentence that would use all those? Give me an idea. How could you use those 3 words in a sentence?
Where they're found in the
Okay, so give me the sentence. Ribosomes, which are found oftentimes associated with the rough endoplasmic reticulum, are essential in the production of proteins. Something like that, right? Something like that. It's not that bad, right? And as you write these sentences, you have to underline the term. So what you're going to do is on the exam, I'll give you these same 11 words back. And you will have already, in your own mind, created these sentences. And then you'll just write them out on the exam. I find that if students can knit these words together in a meaningful way, that they understand the concepts. There's more ways to put these words together than there are people in the room. So I expect each of you will have your own unique way of putting these words together. Now the rules are this: no more than 3 sentences. In other words, I don't want 10 sentences. I don't want 10 definitions. I want you to put these words together. So in 3 sentences, that means that one would have 3 words, another sentence would have 3 words, and maybe the third sentence would have 4 words. Yes, you can only have 2 sentences. That's fine, but no more than 3 sentences. What's a sentence, you ask? Starts with a capital letter and ends with a period, right? So literally, I'll be counting periods. When I turn your test over and I start grading it, I'm going to look for periods. If I see more than 3 periods, something's not right, right? Because there's more than 3 sentences. I'm not going to worry about run-on sentences. I'm not going to be the grammarian here and worry about run-on sentences. But you can easily create 3 nicely worded sentences.
Do they have to be related?
No, they do not have to tell a story. So they don't have to be contiguous or 3 in a row, although you may find that easier to remember, but they're not required to make sense one after the next. Okay, so 3 individual sentences. Now, the words you have not yet learned about in this course: transcription, translation, proteome, genetic code, and mRNA. And we're heading there now. So that's where I'm heading over the next hour, is discussing some of those terms. So be listening for them, and then imagine how you could put these words into a sentence. Think about it. If it's not making sense, come back and ask me about it on Wednesday, and I'll be glad to clarify that for you. There's 11 terms. So the one you don't like, the one that you can't easily incorporate, throw it out, right? You only get 10. Okay, so let's continue this conversation. Again, this content is scattered throughout Chapter 3 in your Martini book, and there's some beautiful demonstrations and beautiful illustrations in there for it to help you. So in this part of the chapter, our focus is on DNA. And we've talked about a little bit about the structure of DNA, and we know that DNA will wrap around proteins to make chromosomes, and we call that material the chromatin that makes up the chromosome, and that the chromosome will duplicate or copy itself or replicate itself at some point in the cell, and we will see those replicated chromosomes appearing sort of like an X. We're not going to go into much more about chromosomes than that for now. I will say, though, that this has become old hat now, but it's hard to find a biology book that doesn't mention Watson and Crick. These are almost dinner table names. Watson and Crick back in the 50s described the structure of DNA. Now, before the 50s, now that, 60 years ago, it's not that much time on the scientific time scale. But in those last 60 years, we have learned so much about our medicine and biology and the overall working of our cells because of what they began. Now, I always kind of give them a hard time. They didn't do any really fancy experiments. They just played with tinker toys, and they played with models until they got the right combination. And they really kind of took information from about 6 other scientists to create this model of DNA. So the other folks should have gotten as much or more credit than they did, in putting this together, but they get the credit, Watson and Crick get the credit for coming up with this model. Crick was his PhD mentor, and Robert Watson was the graduate student, and Watson is still alive and doing well. Now, one of the people that they kind of ripped off, if you will, is this young lady, Rosalind Franklin. Rosalind Franklin and Maurice Wilkins, but Rosalind gets most of the credit here. She had been playing with DNA and playing with X-rays. We didn't know the structure of it yet, but she was getting close, and she was shining X-rays onto DNA. I don't expect you to understand this. I don't understand this pattern. But what it told them and what it told Watson and Crick is that DNA was a helix. Okay? That it was a double helix and that it had a particular diameter. So there was a fixed diameter and it was a helical molecule. Now, she would have also gotten part of the Nobel Prize, but she died very young, probably from playing around with X-rays. Right? So she died, much like Madame Curie, of radiation exposure very young. But they don't give the Nobel Prize to those who've already passed away. So she missed out on that. She passed away before the award was given. Now, we've already mentioned DNA and RNA in passing, just a little bit about what the difference is. Let me dive in just a little bit deeper, but not hugely deeper. So we know that DNA and RNA are both examples of nucleic acids. We know that both of these nucleic acids are made up of bases, and that there's a sugar difference. DNA has a sugar called ribose. So RNA has ribose. DNA has a sugar called deoxyribose. When you look at an RNA sequence, you will see 4 different bases. A, C, G, and U. Versus when you're looking at DNA, you will see RNA in the cell is typically single-stranded, whereas DNA is typically found as a double-stranded molecule. I'm not going to ask you to know the difference here. I did define a nucleotide in the last unit, and you know that a nucleotide is defined as the base, the sugar, and the phosphate group. Okay, so that hasn't changed. The phosphate group, the sugar group, either ribose or deoxyribose, and the base. And the base would be the A, C, G, U, or T, depending upon what we're talking about here. Notice that the phosphate group is negatively charged. And I mentioned to you last time that it's that negative charge on the DNA that helps it to bind to those histone proteins. Those histone proteins in the chromatin are positively charged, and it creates a really strong interaction. Here's your double-stranded DNA. The backbone of this, I always think of this as being like a spiral staircase. So the handrails are what? The handrails are alternating phosphate, sugar, phosphate, sugar. If it's DNA, what's that sugar? Deoxyribose, right? So deoxyribose is the sugar, alternates with phosphates, and then what are the stair steps? If this is the winding staircase, the steps are the bases coming out and holding hands with each other. And it turns out that the bases are very predictable. C, cytosine, will always bind with guanine, G. And thymine, T, will always bind up with A, adenosine. Or adenine, sorry, adenine. Now, what Watson and Crick, this is all going back to Watson and Crick and what they were trying to figure out. And remember I told you that Rosalind Franklin's work told them that DNA was of a fixed diameter. So this double helix was of 1 diameter. So part of the mystery here was that there had to be molecules, thymine and adenine. Now notice that the thymine is a single circle molecule, whereas the adenine has a double ring to it. And part of what their work showed is that there always had to be one single binding to a double. So the A's always bind with T's and C's always bind with G's. We refer to this as complementary pairs. It couldn't be any other way. If there was a T binding with a C, what would happen? Look at it. If there was a T always binding with a C, that would be too skinny. And if there was an A binding with a G, then those stair steps would be too far apart, right? So in order to keep this fixed diameter, the molecule had to be composed of a smaller and a larger molecule coming together. Again, we call those the complementary bases. And in my nomenclature over here, I have the equal sign, A equals T, not equals, but two lines. Those two lines represent two hydrogen bonds. Remember, hydrogen bonds are those weak interactions between hydrogens and usually oxygens or other negatively charged atoms. C binds to G, and there's a third line there. So there are three hydrogen bonds that hold C and G together. So really DNA is a double-stranded molecule only held together, the zipper is really only held together by a little bit of these hydrogen bonds. So this DNA can be pulled apart. Just a couple more terms here. So the purines, it's just a group of molecules. A and G belong to the purine family, whereas C, U, and T belong to the pyrimidine family. The pyrimidines, as you see, are single ring structures, whereas the purines are double ring structures. The way to remember it, what's that? The uracil, which is what was found only in RNA. So again, the U is found in RNA, the T is found in DNA. The way I remember this is I imagine that in making the pyramids, the Egyptians had to cut the stones. Right? So CUT, those three bases are pyrimidines. Just a little word story there, to cut the stones to make pyramids. Question?
How come the A and the C can never link together and the T and the G can never link together? Or can they?
Okay, so I told you a moment ago that we wouldn't want two big ones or two small ones together. You're pointing out something else, the number of hydrogen bonds between them. So remember I told you C and G connect through 3 hydrogen bonds. So the reason that T would not go with G is that they would not find each other very compatible. Okay? So ideally, C with G and A with T. Another way of saying it, one purine always matches up with one pyrimidine. Right? One purine, one pyrimidine. And that's going to give us a diameter which is fixed, right, in its distance from one side to the next. Okay, back to the cell cycle just for a second. And now we're going to focus on this portion of the cell cycle called the S phase at the bottom in yellow. And the S phase, again, S stands for synthesis. So it's during this time that the cell is going to synthesize everything that it needs so that when it divides, it has 2 of everything. The most critical thing to copy is going to be the DNA. So the DNA, the chromosomes, have to be duplicated or copied, and the word for that is replication, to make a replica, to make an exact copy of it. That's the cell's objective, is to make an exact copy. The problem is there's 9 billion of these letters, 9 billion of these bases, and errors happen. So even though the intention of the cell is to make an exact replica of the DNA, as it is doing this replication process, errors will occur. This also means that as we come out of S phase and we're in G2 and going into mitosis, that we've already duplicated everything, right? So that as the cell goes into mitosis and it divides, it already has made duplications, replication of all the organelles and the DNA. So during anaphase, we're just pulling apart duplicated copies. Now, this process of replication brings us back to your question as well as what Watson and Crick figured out. And that is the cell must have some pretty slick way of figuring this thing out. It's not that complicated. You already got it on there. But every time when the cell is replicating, making a copy of the DNA, it looks and it says, oh, there's an A, so I'm going to make a what? I'm going to match it up with a T. And now I see a T, so I'm going to match it up with an A. And down it goes. It's very, very straightforward. And this is the basis that Watson and Crick figured out, but it's also the very way by which our cells quickly copy or replicate the DNA. Now, replication is fast. It's super fast. In fact, it's too fast in some ways. We have to balance quality control here. It's like Toyota or any car manufacturer. They have to balance making enough cars to satisfy the need, doing it fast enough to keep up, but good enough, right, to minimize the errors through the production process. And that's exactly what the cell has to balance. The cell has to get the DNA copied so that the cell can divide and we can have more cells, but it has to make sure that its fidelity, right, or its accuracy is high. In E. coli, a favorite gut organism, right? Invades hamburger, all that. They only have about 5 million base pairs. So we have billions. DNA in E. coli is only about 5 million. And the E. coli can divide about every 20 minutes. You can put bacteria in the right conditions and they will divide every 20 minutes. Well, in order for a bacterium to divide, it must have gone through the replication process and through the cell cycle. That means that a bacterium can replicate about 500 nucleotides per second. That's pretty fast, right? I mean, it's clipping along, making 500 copies a second. We are a little bit slower. Part of the reason we're slower is that we can't afford to make as many mistakes, if you will, if you want to think about it that way. We've got to do this at a rate where we minimize the errors. And we have a huge genome. Genome is the word for all of our genetic material. We have about 6 billion base pairs to copy. But the fastest we can do that is 8 hours. So we do have some cells in our body that can divide every 8 hours, especially some of your blood cells and some of your epithelial cells, the cells making up your skin, the cells lining your gut. You know, if you burn your tongue, within a day you feel pretty good. Why? Those cells have already replaced themselves rather quickly. So some cells can grow very fast. About 8 hours, about as fast as our cells can do it.
I heard that if you like cut your tongue or like cut your like did something, one of my teacher's sons like bit his tongue really really bad, and like the next day I was like here, it was like 24 hours. I don't know. He said something about your tongue can heal like really fast.
It can, right? Your tongue is especially fast. The inside of your mouth is especially fast. Your skin, right? I mean, it's very, very fast. Absolutely. Now, we've got a balancing act here, though, right? Between how fast we go and how accurately we can copy the DNA. So mistakes are going to be made. And in genetics or in biology, when mistakes are made in the copying of the DNA, we call that a mutation. So replication is going to make, on average, a mistake every 100,000 times. Well, that's a great quality control. I mean, if Dell only put out 1 out of every 100,000 bad computers, or Toyota only put out 1 bad car out of 100,000, they'd be, amazing. So our body does amazing things here, but errors do happen. And here's what a replication error looks like. So this is the DNA that starts off. We have G's matching C's and A's matching T's. All of that is normal. During the replication process, which I'm not going to dive into in great detail, something goes wrong such that a G gets bound up with a T. Your very question. Can G and T bind up together? Size-wise, they can, but they're not a perfect fit. So now the cell says, okay, I see a G and a T. Which one's right? Right? Which one's correct? And half the time it will guess correctly and say, oh, I guess that G was correct, or that T, I guess that that G was okay. But other times it'll make the wrong guess. And so now in the next generation, it will incorporate an A and a T pairing. Well, we started off with G and C. Through a mismatch, we get to an A and T. That's a change in the DNA. Therefore, that is a mutation. Now, that mutation may have no effect on the outcome or on this individual or on that cell, or it could have devastating effects. We don't know yet. Okay? But that's what a mutation is. So a mutation can be harmful or not harmful to the cells. Let's say I go to the beach and I sit outside too long and cells in my skin get too much UV radiation, and from that radiation undergo genetic changes and get mutations. And that particular cell begins to grow uncontrollably. I have skin cancer. Is that going to affect my kids directly? I mean, if I die, I guess that affects my kids, right? But is that change in my skin cell going to affect my sperm? No. So that would be considered a somatic mutation. Yes, I can affect my skin, I can affect any other organ in my body, but it's not going to affect genetically my kids. If, however, I sustained a mutation in the process of making sperm, then yes, right? That sperm could potentially pass on that mutation, whatever that mutation is. That would be a germ line or a germ cell mutation. So it's one thing to have a somatic cell mutation. Remember, soma means body. So it's one thing to have a body cell that undergoes a mutation, but that is not heritable. But if my sperm producing or if your egg producing cell undergoes a mutation, then that mutation can be passed on to our children. So a mutation is simply a permanent change in your DNA. Now there's two main types, and you can take an entire genetics course on mutations. So I'm really simplifying this, but hopefully giving you the important highlights here to understand this. Two basic types of mutations. Those which involve entire chromosomes. Well, I described one of those already. That would be Down syndrome. Right? That would be considered a mutation as well. Even though the ACTs and Gs are the same, there's a failure of chromosomes to separate, or there's a problem in overall meiosis. What I'm going to describe to you now are point mutations. And point mutations can come about by two different ways. One, an error in replication. I told you 1 out of 100,000, there will be a mismatch, a problem with replication. Or there could be something in our environment, UV radiation, X-rays, chemical, something that we are exposed to which changes our DNA. So replication errors are problems with the copying versus something in the environment, X-rays, gamma rays, you name it, chemicals, oxygen that we breathe, that change our DNA. So again, you could have carcinogens. Right, carcinogens, we should recognize that. Carc meaning what? Right, carcino, cancer causing. Gen, something that in the beginning forms or creates. Okay, so carcinogenic. Radiation, X-rays, ultraviolet light, sitting on the beach too long. Tanning booths. No, they're not safe completely. So all of those things can be changing our DNA. The good news is our cells are beautifully equipped to fight it off. We have collectively over 100 different ways to fight off mutations. So as they occur, our body can flip it back, switch it back, and avoid the mutation, because we've got to maintain our genetic identity, right? The perfect ideal world is that every cell in my body is exactly like every other cell, and that my offspring would have those normal cells as well. But again, mutations and changes do occur. Now, it's estimated that each of us, on average, carries around 5 or 10 genes, which can cause disease. Right? And it may be causing disease in our body, or it may not be, or it may be that if we carry the same gene as our spouse that our kids would have a greater chance of being affected by some of these. I've already mentioned that all cancer is now considered genetic. There's a beautiful field out there of study called genetic counseling, if you're not familiar with it. And it used to be that genetic counseling was all dealing with patients who have had a child who had some genetic problem, or maybe they have a family history of genetic problems, and they would seek out the help of a genetic counselor to do some prenatal testing or to do some counseling on what are the chances of my child having X, Y, or Z. As much as that's going on now, there's also now cancer genetics. So now you go see a genetic counselor who deals with cancer genetics, and they say, okay, Uncle Herman, he had lung cancer, but Aunt Susie had ovarian cancer, and this person had stomach cancer, and they're trying to figure out, are all these different cancers related, and are you somehow at a higher risk, and therefore should you be monitoring differently than another person? We know that with age, cancer rates go up. We know that. We know that statistically. We know that in our everyday experience. And this graph shows that with age, the number of mutations goes up. And this is for males. And for every 100,000 males, prostate cancer becomes the most prevalent cancer, followed by skin cancer. I don't think lung cancer is on this graph, but lung cancer also goes up. But they all go up with age. And the reason is, we know that the more mutations a cell has, the more likely it will become a tumor, okay, a cancer cell. And these mutations are cumulative. Okay, so a stop at the beach today, and I get a little bit of extra radiation, and now this cell has a mutation. And then that cell divides, and every cell it makes also has that mutation, doesn't it? Right? So that skin cell divides, and every other cell from it, from ever and ever, will have that same mutation. And then next year I go out, and I'm at the beach again, and darn it, that same cell or one of those same cells also gets a second mutation. And we know it's that cumulative effect of all those mutations that can lead to cancer cells. Colon cancer, somewhere between 4 and 7 mutations is necessary to have cancer, or colon cancer. Now, some people are already born with some of those hits. If you have a genetic history, if you have a family history of colon cancer, what it means is that you were already born with maybe 2 or 3 of those mutations. And now you only need to get 2 or 3 more. And so you're likely going to have cancer earlier in your life, and you need to be watching for that colon cancer. Other individuals did not inherit any of those family mutations, and so they're less likely to get it. So your likelihood to get a particular kind of cancer is both genetic, what have you inherited, as well as environmental, what additional risk factors are you exposing yourself to knowingly or unknowingly. That's not a good conversation. It's good information. Now, we're made up of trillions and trillions of cells, and we're losing cells and making cells every second. In fact, you are making 25 million new cells every second. 25 million cells are undergoing the cell cycle, are somewhere in mitosis. So that means that there are a lot of mutations happening, right? Millions and millions of cells every second, copying billions of nucleotides every second. Now, when a mutation occurs, your cell has 3 choices. Let me get through this slide, and then we'll take a little break. But when a mutation occurs, 3 choices. 1, the cell can recognize the damage just happened and repair it. I told you the cell has a lot of tricks to fix the damage. So if it sees it, it's almost like an old-fashioned typewriter. You guys don't know what typewriters are. But in the old days, right, you made a mistake and you pushed the back key and it went back, or you put some whiteout over it and you fix the problem before you go forward. You couldn't fix it once you got to the next row, right? Word processing, you can go back 5 rows up and fix the problem. In the old days, you kind of had to fix the problem quickly or else it didn't happen. So the cell can do that. Number 2, the cell can recognize, whoa, this is too much damage, and basically can commit cellular hara-kiri, right? Commit suicide, apoptosis. So the cell will die, the rest of the organism will survive, but the cell will be so damaged that it will die. Well, that's the good news, isn't it? because that cell no longer will propagate more mutated cells. So we fix it, good. We kill it, good. Or we live with it, right? And we live with the consequences of it. So that change is forever incorporated into your DNA. It's just like your family, right? You either fix them, kill them, or live with them, right? You just get used to them, right? It's the same story, okay? So the idea is that Darwinian evolution, the whole idea of Darwinian evolution is that these mutations have occurred over time, and those mutations have led to changes, and these changes, here we are, we're seeing the evidence of those changes. Let's take a quick break there, 5 minutes or so, and we'll come back and we'll talk about transcription and translation. Moving ahead, but first of all, are there any questions about replication or anything now related to tumors or cancer, anything that's coming together for you? I mean, this is a fascinating area. You could take entire classes on almost every one of these slides, right? Tumors and replication and genetics and meiosis. So there's a lot of information here. What you're responsible for is simply the level that I'm teaching it to you. This is what you need to understand, and you can go deeper if you want to, but it's not something you need to do to be successful, okay, on this test. Now, the next thing I want to look at, we talked about replication very, very quickly. And replication is the copying of the DNA, and that in the copying of DNA, mutations occur, replication errors occur. We've talked about that. Now let's talk about transcription and translation, and the making of proteins within the cell. We know that DNA is the control molecule, and that DNA is going to dictate everything else about the cell. And there are two processes here that we need to look at in making protein. So DNA, and where in your cells do you find DNA? Where are your chromosomes? Where is the DNA? It's in the nucleus. In a eukaryotic cell, you are a eukaryotic organism. In a eukaryotic cell, if you find DNA outside of the nucleus, something's not right. That cell is very ill, that cell is dying, that cell is in the process of apoptosis. So typically, normally, DNA will only be found within the nucleus. The first process here is transcription, to transcribe. So if you hear that someone has transcribed something, they have done what? They've rewritten, or not exactly copied, but they've rewritten it. That's a good way of putting it. And in the process of transcription, the cell makes mRNA. Now, it's a kind of RNA, messenger RNA, M for messenger. But we see that we're going from DNA to a molecule of RNA. It's still a nucleic acid. It's still largely the same information. It's still A, C, Gs, and Us, and Ts, right? It's still in the same family of molecules. And then the mRNA will undergo another process called translation. And that translation process will now create proteins. So when you translate something, what do you do? Or what do you tell Google to do? Right? I'm going to go from English to Spanish. And I'm going to change, I'm going to change a lot of things, right? It's now a completely different language. I'm translating it into a different language. So I'm going to break this down in a very global 30-foot view. We're looking down, just try to get an overall view of this. So we'll start off with transcription. Transcription, again, is going from DNA to RNA. Now, to understand some of the challenges here, we need to make sense of our DNA. Turns out that only about 1.5% of our DNA, a very small percentage of our DNA, is known to code for or to be directly responsible for the making of proteins. That is, if I had your DNA and it was stretched out over a 6-foot long string, only 1 inch at the end of that 6 feet would actually be critical for making protein. The other 5 feet 11 inches would be, quote, junk. It would not be important. Now, we're learning that some of that junk is important, but I'm going to keep the story simple. So much of our DNA is junk, or another way of saying it, it is non-coding DNA. It does not directly code for or give direct instructions for the making of proteins. And the question is, well, why do we have it and where did it come from? And those are outside the scope of this course. But we'll refer to it as junk DNA. And much of that junk DNA are introns. I'll introduce introns in a moment. And there's also been a lot written about repetitive sequences. Much of our DNA is made up of these repeating units which seem to have no direct purpose. And we have some ideas as to how and why they're there, but they don't really give us any direct advantage. In fact, if you match our DNA up with other primates, there will be 10% of our DNA which seems to be very, very similar, but has no purpose that we can identify. So the DNA has a lot of junk in it, and the cell has to figure out what's junk and what's not junk, what's important and what do we need to get rid of. So as the process of transcription is occurring, as the DNA is copying to make RNA, this is where the junk will be gotten rid of. And we say that the RNA is going to become edited. It's going to get rid of the junk. And it's going to cut out these sections called introns. So the introns are cut out. It's going to keep the exons together. Now, if I were naming this, I would have flipped the names. Right? I would have gotten rid of the exons and I would have kept the introns, and I didn't make the rules. So look at those words. They don't make sense at first glance, but the introns are gotten rid of, the exons are kept. In this diagram, the introns are in purple. That's junk, stuff we don't want, and it will be basically cut out of the DNA. It literally makes like a little lasso kind of structure and cuts it out, and poof, it's gone. So now what we've done is we've linked together the exons. So the exons become linked together. The good stuff, that 1.5% of the DNA that matters, gets linked together. The rest of it, the introns and all the rest of the junk, is gotten rid of. And what started off as DNA, now we have messenger RNA. So messenger RNA would be that final piece of RNA where we've gotten rid of all the junk and we're holding on to now just the good stuff. Just to give a little bit of a lesson, historical lesson, it was thought when I was in graduate school that the human had somewhere around 100,000 genes. And the reason it was thought that we had 100,000 genes in our genome is that there were known to be about 100,000 proteins. Oops. There was about 100,000 proteins. It was thought that every gene made one protein. And that was just the calculus. That's what we thought. We thought that every gene made its own protein. And then, as you know, there's this huge thing called the Human Genome Project, right? And we were able to sequence and figure out all the ACTs and Gs in our genome, in our body. Along the way, though, if you look at different textbooks through the last 20, 30 years, you'll see it go from 100,000 to 80,000 to 75,000 to 65. And the number now is somewhere, you'll see different numbers, certainly less than 30,000 genes. We still have 100,000 proteins, right, in our body. But we now know we have a lot fewer than 30,000 genes, some numbers as low as 18,000. How does the cell do that? How do we do this? Well, it turns out that one gene, this represents a gene, one gene can rearrange these exon units in different ways. So this is one gene that can make a protein by combining sections 1, 2, and 4. creates this protein. But if I reconnect sections 1, 3, and 5, I can create a very different protein. Another way of saying this is that our genome, all of our DNA, is less than our proteome. Now, that's a fancy way of saying genome, all the genes in your cells, proteome, all the proteins in your cell. So we now recognize that it takes just a few genes to make a lot of proteins. If you look back, you'll see that word proteome in that list of terms. Okay, so proteome refers to all the proteins that your cell has or all the proteins that your body possesses.
When they're different like that, like 1, 2, and 4, and 1, 3, and 5,
Yes.
what happens to the 2?
Again, they're cut out, just like the introns.
Okay.
Just like the introns were cut out, so too some of the exons are cut out. So we call this splicing or alternative splicing, and we create vastly different proteins. Hang in there. I know there's a lot of concepts that need to come together. So transcription then is taking information from the DNA and making a molecule called mRNA. As the name suggests, this is a messenger molecule. It is going to carry the message from the DNA to the cytoplasm. It's a relay molecule. In this situation, the nuclear DNA, right, the DNA in the nucleus is the template for making mRNA, and this process has to happen in the nucleus, right, because that's where DNA is. So if you're going to make mRNA from DNA, this better happen in the nucleus because that's where the DNA is found. So let's take a look at a cartoon picture of this. We're inside the nucleus. Here's just a simple little molecule, a little bit of my double-stranded DNA. What's not shown here is that the molecule of the double-stranded DNA molecule will open up. And there's a lot of enzymes involved here, but as a result, some of this DNA will be copied or, quote, transcribed to make this single-stranded mRNA molecule that's shown in yellow. We're in the nucleus. This is happening inside the nucleus. In fact, this would be the edge of the nucleus over here. So that's the process of transcription. Let's take a look at it. The next step is to take that mRNA and now make out of it protein. So there's really two processes that have to happen here. First, we have to have transcription, and then we have to follow it with translation to make the protein. Now, this is exactly what Watson and Crick talked about in their paper. Right? Not only did they create a tinker toy model of DNA, but they also proposed, based upon that model, that DNA must code for information necessary to make proteins. So they described what's called the central dogma, or the idea that DNA goes to RNA and then goes to protein. So DNA to RNA to protein, and that is this idea of the central dogma. So let's zoom out just a little bit and take a look at this overall process. Same picture. DNA, we're in the nucleus. We undergo transcription to make mRNA. Do you remember that the nucleus is surrounded by an envelope, but that envelope is double-stranded, or double-layered, and has some pores in it? Right, there are big gaping holes in the nuclear envelope. And there are a lot of things that have to go in and out of the nucleus, but one of them is these very, very large mRNA molecules. So when you first make the mRNA, it has to squiggle through the nuclear envelope, the nuclear membrane, and get out to the cytoplasm. Now, what's it going to find out here in the cytoplasm? What is usually right next to the membrane, to the nucleus? The rough ER, right? A bunch of membranes which have on them what? Ribosomes. Now here the rough ER is not shown, but it does show you some ribosomes. And this big purple guy here is a way oversized ribosome. But for the picture, it's there. And what's that ribosome going to do? That ribosome is going to be the major organelle required to translate, to take this mRNA message and translate it into a protein. Ribosomes are only found out in the cytoplasm, so the process of translation must also occur out in the cytoplasm. This green that you're seeing, there's your protein, okay, that's been created. Here's another picture of the same process, different textbook. In fact, weirdly, the same colors. So we're inside the nucleus. We see the double-stranded DNA. And what would you call step number 1? I'm going from DNA to mRNA, so I would call that transcription. Absolutely. Step number 2. I'm just going to call it processing. Now, what does that mean? What's happening during that processing? That's when those introns were being spit out, all the junk was being taken out, right? And we end up with our final mRNA molecule. So number 2 is the processing step. And then number 3 would be what? Yeah, so 3 is translation. Okay? So just keep it that simple. If you've taken a general bio course recently, you probably had more detail than this. I'm trying to keep it at just an overall understanding. And then when I get to the end of this, which may not be until Wednesday, I think you'll understand why we're going through these steps. Now, translation then is the second process. Let's look at the words here. Translation, we're going to take the information from the mRNA. It happens where? Out where the ribosomes are, that is out in the cytoplasm. How did the mRNA get out there? It had to travel through those nuclear pores. That mRNA is now going to link up with some ribosomes. Now, those ribosomes could be floating around in the cytoplasm by themselves, or they could be bound to the endoplasmic reticulum. Reticulum means web or a network. So this mRNA is coming out of the nucleus and is being caught up in this web, caught up in this network of membranes. Now, there's another player here. There has to be tRNAs, and I'll show you those in a moment. The transfer RNA are the molecule that's going to bring the correct amino acids to the ribosome. And the ribosome is then going to take those amino acids and link them together to form the protein. Now, these two words do come in alphabetical order. So transcription does alphabetically come before the word translation. And I wish they were totally different words, right? They're so similar that they cause confusion. But if you're really, if you're still having trouble with this, just remember transcription does come alphabetically before translation. Another way of thinking about this is here, DNA, right? DNA undergoes the change to mRNA. Now, it's not like hocus pocus, the molecule changes or becomes, but from DNA, mRNA is created. We're still dealing with nucleotides. We're still in the same alphabet. We're still dealing with A, C, G, and what? T here. And in mRNA, we have A, C, G, and U. Other than that, though, we're still dealing in the same language. We're still dealing with the language of nucleotides, or the language of nucleic acids, and the alphabet, right, would be the nucleotide bases. But then when one undergoes translation, there's a huge change, because now we are changing alphabets. We are going from A, C, G, and U to amino acids, and there are 20 different amino acids. So we're going from a system, an alphabet, a language with only 4 letters, A, C, G, and U, to an alphabet or a language that has 20 different characters. It's a major translation, right, in the overall process. Let's take a look at this artist's rendering of the process. There's a double-stranded DNA. We see transcription occurring, and we make our mRNA. Okay? Single-stranded mRNA. The mRNA is going to squiggle through those nuclear pores out into the cytoplasm, where it'll get bound up by endoplasmic reticulum or run into a ribosome. The ribosome is shown here as this big purple dude. Okay? What we see is that this mRNA, we see 4 colors on it: green, blue, yellow, and orange. Those green, blue, yellow, and orange represent in no particular order A, C, G, and U. And you'll always see it matching up. So, orange always matches up with blue. Well, what could that be? Orange always matches up with blue. That could be what? A and U, or C and G. Remember, there's no T's here, because T is in DNA, and what we're looking at here is an RNA molecule. So there's U's instead of T's. So A's would bind up not with T's, but with U's. A's and U's will go together in RNA world. Okay. And then the green and yellow would be the other pair, whatever that is. The little circle balls, little spheres over here on the left-hand side, those represent some of the different amino acids. And this strange-looking lunar molecule-looking thing, that's a tRNA. And we're going to see the tRNA is going to carry, shuttle, bring those amino acids over to the ribosome, but it's going to know which one to bring based upon a code. Then as the ribosome is reading the instructions on the mRNA, these tRNA molecules are coming in, and they are specific for that sequence. So each tRNA matches up to 3 different nucleotides on the mRNA and carries with it a particular amino acid, which then is linked together in the final process of translation. So just another picture of this. Here's the ribosome, the single-stranded RNA. This looks like a piece of a puzzle, right? That's the tRNA, and that tRNA will always carry with it a particular amino acid. That means that there are different amino acids, and there must be, therefore, different tRNAs. So the genes, the DNA on your chromosomes is described as having a genetic code. We've heard that term. That's one of your 11 terms as well, genetic code. Now, genetic code has a couple different meanings. It's okay to say that your cells contain your genetic code. But we also see that the cells have a system, a decoding system, which is also referred to as the genetic code. Let me explain this, and then we'll be out of here today. So on the top, and this is kind of getting to the bottom line of why we care about replication and mutations in DNA. On the top, the red is DNA. I know so because it tells me. I also know so because there are T's in here. Whenever you see T's, you know you're dealing with a DNA molecule. You never see T's in an RNA. It's only 1/2 of the strand, so it's been separated. We're looking at one strand of the double-stranded DNA. In the process of transcription, the A, C, T's, and G's were transcribed to give us the single-stranded mRNA molecule. Here, rather than T's, we find U's. How did the cell know what to do? Just like in replication, it knows that when it sees a G in the DNA, it does a complementary base pairing and gives me a C in the mRNA. An A will match up with a U, or put a U into the mRNA, and T's with A's. So the whole genetic code is based upon the language of 3. Each 3 nucleotides is part of the highlights for kid decoding ring, right? So there's a way to decode this genetic information. And then through the process of translation, the ribosome is able to understand that every time it sees UGG, for example, every time it sees those 3 nucleotides in a row, it will always add the amino acid trip. And every time the ribosome sees UCA, it will know to always add in the amino acid ser. Now, you're not responsible for all the different amino acids, but I wanted you to understand that if there's a change in the DNA, what happens? A change up here in the DNA can cause a change in the mRNA. If there's a change in the mRNA, then there can be a change in the amino acids. And what we'll see is that if a protein changes its amino acids or loses amino acids, it can have devastating effects on the overall well-being of the cell. So a mutation up here, right, a change in your DNA can have dramatic effects on your proteins. That's where we're heading, and we're almost there. We're almost to the end of this conversation. What I'll be doing over the next few minutes together on Wednesday is describing to you the overall genetic code, and you will learn how to decipher the genetic code, and we'll learn a little bit more about mutations and why we can have glowing kitties, right? So that's coming up, why we can have glowing kitties and glowing plants as we talk about genetic engineering. There's not a whole lot left to this conversation. Please go back and look at Mastering Homework 3 is out there. Look through your book. Come back with any questions. And then we'll finish up this information and get going into histology. Now, histology you've already seen in lab. So I'll be going through a quick whirlwind through histology and tissues, and then we'll finish up next Monday with skin. And your exam will be on cells and organelles, today's conversation about transcription, translation, genetic code, DNA, tumors, next time on histology, and then on skin. And that'll make up our first exam. So things are going to be moving along a little faster now. The biggest break was the first 3 weeks, if you will. And now we're going to have something going on exam-wise almost every week between lab and lecture. So hang in there, stay with me, stay current, ask questions, and we'll get through this together.